now it's on. Now it's on. All right. Okay, but you heard me. I'm sorry for the webcast guys who didn't hear me, but well, it's all here in the picture. So, and finally, there's also physical space in basically the space in which the odors travel from the source to our nose or to the noses to the antenna of insects, and there's turbulence and all these different things that are going on, and all these. In my opinion, all these aspects have to be considered if we want to figure out olfaction, because I think we will not solve olfaction by only looking at the brain. All right, to give you a bit more of a concrete example, if we compare, as also Brian did earlier, so I'm picking up basically what you said in the last part of your presentation. If you look at vision and our progress in vision research and how to understand vision, we know a lot about vision. We know a lot about the stimulus space. We know about color, what defines color. We know exactly how to manipulate color and to model color in images. We know about contrast in images. We know what it means when there's contrast between two different pixels or the contrast of a whole image. We know how to modify it, how to modulate it. We know even the statistics of natural images. So this famous paper from Olshausen and Field in 1996, they simply did statistics of natural images, and what they found is if you want to efficiently encode natural images, what you end up is these kind of like basis functions which look pretty much like receptive fields in the visual cortex in V1. That makes perfect sense, so that's a pretty strong case that the brain is actually efficiently encoding its natural stimuli, so efficiently encoding natural images, which is supposedly has evolved to do so. In olfaction, we don't know much about all these things. We know very little about, like, for example, when you speak about contrast enhancement in olfaction through lateral inhibition, then there's an assumption that we know what is to be enhanced and what is the pattern, what is the image that is to be enhanced. But we don't know. We don't even know how many receptors are active in the mouse, and we don't know what is the pattern that is to be enhanced and how to enhance it. What is the goal of that enhancement? And well, these are all open questions which I find very interesting, and I'd like to discuss more during this workshop. Any questions from you, maybe?
The next person I put up was me, and I wanted to talk about odor transduction, some work in my lab recently, and we all know the stimulus space can be very turbulent and complicated. An odor coming off a flower and a bee or any animal trying to move up an airstream to locate that odor source isn't going to be hit with constant odor, but rather they're going to be exposed to these little wisps or filaments for just a few milliseconds at a time. The problem for an animal then is to use this information to effectively locate a source, and we've all seen pictures of dogs and moths flying back and forth using this elegant behavioral strategy for isolating an odor in an airstream like that. It's more complicated, though, by the fact that if you have a target odor here with its own pattern of turbulence and you have a background odor behind it, the wisps of this background odor are going to be overlaid and intermittent with the wisps of the target odor. So you have to do this foreground-background kind of segregation that some colleagues and Thomas is going to talk about here a little bit more in a few minutes. But the gist of it is that in an animal at any given distance, 400, 200 millimeters, 100, and if you're looking through time, how is it going to be exposed to the odor? You're going to get these very transient exposures for just a few milliseconds. Now, something Jim was alluding to earlier when he talked about the mucus layer in the nose, and you have a similar sensillum lymph in an antenna. As you know, the dendrites of a olfactory receptor cell in an insect project up into these hairs that are covered with pores where the lymph, aka mucus, is exposed to the air. What does a ligand have to do? It hits an antenna or gets near the mucus, and there's a boundary layer of quasi-dead air. Needs to get across that dead air by passive diffusion. needs to get to the surface, the aqueous surface, where the hydrophobic molecule has to be latched onto by a odor and binding protein, and carried then by diffusion, presumably across the lymph to a receptor where the ligand is released and can affect opening of an ion channel directly in insects or gating of a G protein coupled receptor in mammals. So I was listening to a talk by someone who studies these odor and binding proteins, and I said, well, how long does that whole process take from boundary layer to depolarization? And he said maybe 40 milliseconds, which I thought was just an eternity, because 3 or 4 synapses into a bee brain we're seeing latencies of 85 milliseconds. Thought that was far too long. I happened to have 2 very capable postdocs in the lab at the time who took it upon themselves to estimate how long it takes using electroantennogram recordings. That's a bee antenna. We did this on many different insects, but they could calculate exactly when the odor got to the antenna using a special device they constructed. So if you now look at when the odor is getting to the antenna, which is represented by this red line right here, well, actually, it's this line you can't see. It's right about right here. And the black line is the electroantennogram, evidence that the antenna is picking up the odor. We estimated recently that that whole process takes on the order of 2 milliseconds to effectively get to depolarization. That's something, a honeybee flying upwind, if you calculate the bee's speed relative to the wind and the thickness of these average filaments, you'll find that the bee's exposed to it for maybe just 2 milliseconds at most. And if you do some back-of-the-envelope calculations of how long do these diffusion processes take, if it's all passive diffusion, 2 milliseconds is right at the boundary when the first molecules would possibly be getting to the receptors, if it's all passive diffusion. So that's what we're... so these processes can be very fast, at least what takes place in the lymph.
Well, thank you very much, Brian. So you set me up perfectly to just introduce this piece. Basically, I brought 2 slides that are not necessarily about my own work only, but it's about the collaboration I do with Paul Siska there and other people in Constance. And you would have noticed that Paul Siska was also one of those postdocs in Brian's lab. He's all over the place with this story about the fast perception of odors and the consequences that might have in the situation that Brian showed so nicely with the plumes, where you have mixtures of odors from different sources, and the different sources might already be emitting mixtures, or typically do emit extra mixtures of chemicals that make up the odor. So you have some situation of mixtures, and it's an open question how animals can make sense of this kind of complex odor environment. So this goes a bit beyond the more fundamental question of what is odor space at all to, well, if you have a really realistic environment, let's say I come in the morning into a coffee shop and they're also doing fried bacon and eggs, then I can distinguish that, even though the bacon and the coffee will be mixtures of at least like tens of components that in principle are beyond the threshold of detection. And I know it's coffee and it's bacon, not some third type of odor that's a coffee bacon. Yeah? And this kind of thing, this kind of question is what we're after here. And as you've seen in the movie, there could be some help from physics in terms of plumes being turbulent, and the odors from the coffee and the bacon don't come at all at the same time. They come with little tiny delays. But then you calculate the delays, and they are tiny. Yeah. So that's the kind of area we're working in. Now, Paul has done experiments with bees where he was looking at behavior, and he could show that in these experiments, when he gives them mixtures that have these tiny delays, they can actually distinguish them. The experiments go like this very quickly. He trains them to respond to some odor A, and that's typically something like nonanol or hexanol, or other related odors. And then he tests them, so they will start responding to this because it's paired with sugar. They respond with the proboscis extension reflex. And once he's trained them, you can see there's some bees that learn and some don't learn, but the ones that learn, they respond with 100% of the time after only 3 trials, and that's about 60% of the bees. If he then starts testing them with these mixtures, And these are to scale, so he tests them with really tiny delays in the mixture. It's almost impossible to see, but there's a 6 millisecond delay here on the 800 millisecond stimulus. He tests them, and then he sees if this stuff comes together, the bees don't generalize that well from what they learned to the mixture. Well, they do, but it's not as much as if they are not together. And there's a significant difference here between when the odors come with a delay, and it doesn't matter which way around the delay is. Okay? And the surprising part here is that what's in red, yeah, it's only 6 milliseconds delay, and they can do it. They can do it even better if the delay is longer, obviously, but the surprise was that it can go down to 6 milliseconds. If you now put the modeler's hat on again, 6 milliseconds is barely enough to have actually a spike in these neurons. I mean, the spikes are easily 2-3 milliseconds wide. Yeah, so we're talking about delays of the order of a spike width, which is tiny. Yeah, and the question is, how can they even make any sense of this? I didn't bring along, and we did then a paper on this in terms of making a first model. I didn't bring along that model, but what I brought along was a bit of an illustration that this is not just something that the bees do, and it's not something that only in olfaction. And I always like to show this illustration of that, which is in the auditory system. So I'm going to make a little psychophysical experiment with you now, to show you that these effects, they exist in different modalities, in different ways, where we do make sense of the environment based on delays. So this example is taken from the literature, the paper's down here, and it involves just having a mixture of tones, and I'll play them to you in a second, and I'll show you that if you leave out one frequency for a moment and play it in later, that the percept changes, and in a way that, if you think about it for a while, it probably makes sense, but for me it was a real revelation because it doesn't reflect the physics really, but it reflects more the sense of things. So let's see whether this works at all. Is the sound on? Let's try that. I think it will be loud enough. So this would be... Sorry. So this would be the fully coherent picture here. Can I make a quick show of hands? Was that one or several tones in terms of, it's a mixture of different tones, right? But how do you perceive it? One tone? One tone, okay. I think we all agree. I mean, it's just a quick test. I mean, if it's a mixture of things that come on together, then it's perceived as one percept, as one object. Now, if you do this thing where the one tone comes a bit later, let's see what we hear then. Okay, any difference? Anyone? No difference? Okay, it's not loud enough. So too bad. Well, do it later again, right? Yeah, I tried, but it's not picking it up very well. It does depend on volume. It does depend on background noises and things like that. But typically, if you have the right conditions, you will hear two tones here. You will hear like the original tone is almost unchanged. You can't see really that one here, that this one frequency is missing. And then when it comes in, it sounds like a second extra tone, like a second extra high tone.
The Mac suppresses that. The Macintosh suppresses that.
It doesn't. I heard it, but it's just not. It's not good enough for the audience. You trained for that. Yeah, maybe I am. Anyway, so story made very short. Brian's already in my knee, breathing down my neck. But the story is basically that just based on it being a bit later, we know it's two different sources now. It's something different, and that's what we think that the insects and we as well do with the odors as well. Yeah.
Okay, well let's if we can just keep moving. So next is going to be Ramon. We're moving into the bad word coding.
Yeah. Anyways, so how I got into this experimental side, but I have both things. So basically what we have been doing for the last 4 or 5 years is building artificial sensor arrays. And we just start looking at this problem of trying to detect gases in real environments using things that you can use in the industry. Then you are faced with these 2 questions. What is the problem you want to solve? That's basically the same as the olfactory system eventually has to do in the real brains. And basically we are trying to solve the problem of classification which is odor identification and discrimination, and regression, estimation of the odor concentration. So the time series that you look in here is that the recordings that we got in the wind tunnel using this artificial sensor array that actually Michael is analyzing now for different gases. And what you can see here is actually the problem of what I'm trying to summarize is that the problem of classification somehow is easier than the regression one. The classification, typically some of the sensors have a very strong response for different type of gases. But while you want to, this is for different distances when you put these sensors at different sensors from the source. But when you're trying to identify the concentration levels, what you have is what Brian already showed in his talks and also Thomas and others, is that you have this turbulence. And this is the main mechanism for information transmission. So it's a random process by which you reach the sensors, and the estimation is obtained by trying to get the random hitting of the plume into the sensor. This is, for example, the photoionization detector, and counting just the number of particles that reach the target. So we have, from the perspective of an engineer and solving the detection of gases in a wind tunnel, you are solving these two problems, classification and regression. And I see many of you guys are discussing a lot the details about how the circuits are designed, but I think it's important not to lose the perspective that eventually the brain somehow, somewhere, has to solve these two problems. And the summary is that one thing is to discriminate the gas, and another completely different is the odor concentration. How do I get to the next one here? So the way we try to sell this for the funding agencies is that we are building these new chemical sensor arrays, but we had these bio-inspirations. So we, as much as we can, we're trying to import what we know about the architecture of the brain, the insect brain in particular. And this is due to this long-standing collaboration I had with Brian, we had with Jill in the past, Maxime and Michael and many others in this room. And basically, we have built models for the classification based on the mushroom bodies of the insects for quite a long time. This collaboration with Thomas, right? And one of the questions that still we are not able to address very well is just how do you actually do the regression, right? What are the circuits involved in the regression? And as far as I know, in terms of the modeling, there's only one paper that deals with this, right? It's yours, Michael, right? Do you have a paper about solving the odor concentration, right, in the antenna lobe? It's not. Is that true? Yeah. So here I just summarize basically the models that we have. These are using just spiking neurons, and the main idea is that this is something that many you probably know already, but the Granule cells in the mushroom bodies are coding. They have this sparse coded representation. But what, in terms of a model, when you're trying to solve a pattern recognition task, a classification task, the place where is the location where you can actually solve the classification task more effectively happens to be in the mushroom body lobes. Why? Because there's this convergence of information going from the KCs that are very sparse into the output neurons in this layer. So when you try to implement computational models and solve a classification problem, and you're trying to identify what areas are more likely to be responsible for solving the classification, it happens that here in the output neurons are the ones that, the output neurons on the mushroom bodies are the ones that are more likely to solve the classification problem. Now, the second problem, the regression, is much harder to have because, of course, you need to be able to call odor concentration and having some brain center capable of estimating the concentrations, right? When you're trying to solve the classification problem, something that is very important is to have a gain control, right? And then you need to find some other brain circuits capable of coding for the concentration. And one of the hypotheses that is actually posed by Tiago Mosqueiro, one of my students, is that this group of neurons, the feedback inhibitory neurons that go into the mushroom bodies, are capable for coding for the concentrations. These are some models that we were able to do. So anyways, to summarize, we have models for solving the classification problem. Now we are attempting to solve models of the regression via the feedback neurons, which are known to be very important in the mushroom bodies.
Yes, no, just
Okay.
on the keyboard.
Okay, thank you. So to be a little bit provocative, so we want to have discussions later on. Brian and Michael stressed this example of visual perception, and I would jump into this and say, well, so that's the problem if we are modeling the olfactory system just like you would have models of the visual system, and so the visual system as a system is very well studied. You have the neural circuits, but what you really understand there is basically the hardware of the olfactory system, how it's running. So we're, painting circuits on our whiteboards. So, but the problem is if we want to understand what the biological system is actually doing in this task, or how evolution tuned the olfactory system, we have to focus more on like the software. So what is actually the problem that the system is solving? And doing so, is the microphone off, right? Okay, okay. So what the olfactory system is actually processing, like in the visual system, are things that we just don't understand in the visual system either. So, I mean, you have chairs in different orientations and you see them all being chairs. But nobody from the vision community can tell you what are the neural correlates of, just transferring signals from the visual cortex or from the retina into something that is for you a chair in any direction. So this is just not solved. And so we want to address this in looking at, kind of like basic understanding of what the system is processing. So this is more like my microscopic view. And I mean, you might be all familiar with this picture, which is kind of like a comic of what we expect. So there are some receptors and, some chemicals have some features, and Michael was also addressing this, look at natural image statistics. Well, one single odorant is kind of like a natural stimulus, because, I mean, this is a compound that comes in nature and maybe in a complex mixture, but still, every single odorant and every single molecule that we are perceiving is already a mixture of some receptors. And, these 3 receptors that might be evolved in the perception of a single odorant is not, activated by a single molecule, but, by 3 molecules of the same type, activating these 3 receptors. So, there was a recent work in 2014 by Castro et al., and they analyzed, olfactory categories, and they found out that there will be like 10 categories where we categorize our odor, sensations. These are verbal descriptors, but we wanted to have a similar picture on the early sensory input in the system, and for this we took the rodents data from Michael Leon's lab, where we have, from my point of view, still, even though they're ignoring the temporal dynamics, the only input pictures of single odorants that really cover the whole bulb. And we did a very similar analysis to the Castro data, so we also did a non-negative matrix factorization, and the idea was there, take the single odorant images and extract kind of like basis functions that explain this neural code, or basically the functional patterns that drive this code. And we came up with, well, it's a wrong order, let's see. So we came up with like an alphabet coding over the whole bulb, inducing 9 odor words, or like letters in this coding alphabet. And this corresponds very well. So these 9 rodents alphabet corresponds very well to these 10 categories they found for human verbal descriptions. But there is not such a thing like one of these words correlates to, or one of these activity patterns correlates to an odor quality. Because we have no categories here, but if we look at our coding patterns, we need mixtures of 2 to 3 of these patterns to, reconstruct the actually activation pattern that can be seen under stimulation of a single odorant. That means basically that one of these categories that we perceive is basically an evaluated function coming from, a mixture, a real odor code. And I think it would be interesting to learn how these odor codes are transferred into these categories that we describe odors to. So I think when I run out of time, then everything else we can discuss later. I mean, maybe the final thing, for us the question is, what are actually these patterns mean then, right? So what's the meaning of these patterns? And I hope that I will learn this talking about these models. Kind of like learning not only about the hardware and software, but also about the middleware. So, okay.
Thank you, Amir. Okay, well, let's move on. Diego's next.
Yeah, so I'm going to bring in the awake behaving animal and the quote-unquote mitral cells, and the quote-unquote is important. And I'm going to start with basically what Christian Linzer was saying, that what we were thinking at the time, and I was coming in from the last talk, in fact. I was doing work trying to understand what those odors look like in the glomerular layer. And the question was, is there an enhancement of contrast? And the one thing that I remember the most is the work of Mori in the anesthetized animal. Then we went to the awake behaving animal. And what we found, which was published in 2008, was that when we had the mouse doing a go, no-go, where basically when odor is rewarded, they lick for it and they get water. And the other odor is unrewarded. So you have S plus rewarded and S plus, S minus unrewarded. And you have these blocks of 10 trials for each. And what happens is at the beginning, they have no idea what each odor means. And they're at 50% correct. And then they learn. And what we were expecting to get was basically responses as a firing rate in the mitral cells that would be different in different mitral cells. But in general, what we found was that in the first block of 20 trials, there was no response. There's a very small percent of cells that do respond differently. And then there was a development of a response that was, in general, excitatory for the rewarded odor and inhibitory for the unrewarded. And when we flipped it, it went backwards in a large number of cells. So this one that was responding as an inhibitory response would now respond as an excitatory response. And that was very unsettling. And basically what we've done is repeated the whole thing. So now we have basically done 251 units, 33 sessions, done exactly the same experiment, but we have added something to it. Now, so let me go through this. Basically, this is the forward go, no go, where you have odors A and B, and A is S plus, B is S minus. Then you flip it to a reverse, so the animal is somewhat confused the next day, and basically they start responding backwards. And then they figure it out, and they actually do fairly well. We had done that before. Now we do a go-go, where basically you give water to both. And of course, they stay at 50% because you get water no matter what the odor is. Some of this is basically the same thing as what we had before. If you look at the percent of cells that were divergent like this, you start basically, the interpolation is to 0, with 251 units. And then you go to about 8% that are divergent. Now, this is statistically divergent, and I'll emphasize that, and statistically, quote unquote. And then you go to the reverse go, no go, and you start very low, and eventually you come fairly high. Now, in the go-go, you are flat. So it's a small number of cells that are statistically different, but it's fairly flat. It does not change. Now, the question is, so this is really bringing reward, information and reward, but those cells likely are bringing information, identity or intensity, and how do you pull them out? So then we went to things that Friedman had said, and that Leslie Kay has done, and many people have done, which is this system resonates in the gamma frequency range. So this is now optogenetic stimulation of the input, but Isaacson has also shown that if you stimulate piriform, you get the same resonant frequency. And this is the gamma field potential. And you see this beautiful resonant circuit, which actually happens at the gamma frequency. So if we look at the spikes in the mitral cells and we try to see if they're coherent with these, and there's a coherency measurement, what happens? And the result was fairly interesting. So the first thing that I'm showing here is basically the spike rate. So I'm just redrawing what I had before. So what I had before was that. And I'm just redrawing it there. And now we do the coherency, and I'll put it in there. And so basically now we're looking at whether there's a coherence between the spikes, and if we were in the box and we were smelling the odors, we would smell rose at the beginning, but we wouldn't know what it means. And what we find is even in the first block, there's a very large response that's basically flat as the animal response. And if you do a decoding analysis, even with about 10 of those units chosen randomly, you can actually get an algorithm to say that this is the S+ odor or the other odor. So that brings information about some feature that does not change during learning. It may be identity, it may be intensity, we don't know, we didn't classify them very well in that sense. And that's basically.
There you go. Unless you want to talk about alcohol.
I have a slide that looks almost exactly like that from before. So, I'm really interested in mitral cell versus tufted cell differences. So I loved the discussion we had earlier. If anybody wants to talk about that more, it's one of my favorite hobbies to debate mitral cells versus tufted cells. But what I wanted to show here is just some recent data, sort of some summary figures from a manuscript that's submitted that I think will be of pretty broad interest to this group. So we know that granule cells, and this is what all this study is about, are critically important to olfaction. They're involved in decorrelating mitral cell responses. Arguably, they're certainly involved in temporally patterning mitral cell responses. So granule cells are critical, but we don't really know how sensory input recruits granule cells, and so that was the main topic of this study. So this was all in slice work, and it's really looking at granule cell inputs downstream of activation of a single glomerulus, and we'll get into activation of multiple glomeruli. And you can see that here I'm using optogenetics. I use a variety of approaches in this study, but in response to a single optogenetic stimulation of a single glomerulus at a holding potential of -60, so just looking at the excitatory responses, you see what everybody else has seen in the field. You have asynchronous excitation, and that's shown in the blue there, onto this granule cell. But I just did a very simple thing. If you depolarize the cell to the reversal potential for excitation, now looking at synaptic inhibition, you actually see that you get very closely time-locked feed-forward inhibition onto granule cells. This is just one example, but it pretty much found it in every single granule cell that I recorded from. Granule cells receive rapid, highly reliable, synchronous feed-forward inhibition. I think this is a critical circuit component that hasn't been looked at that can have a lot of ramifications as far as beta versus gamma oscillation generation and how granule cells, the patterns of activity that they generate. More interestingly, if you then do multiple glomerular stimulation, or if you move your optogenetic stimulation into different glomeruli, as I've done with this example, you can see that granule cells not only receive excitation from multiple nearby glomeruli, this is all very local, all neighboring glomeruli, and that's shown here. These are all uniglomerular, one glomerular activations. You get excitation onto this granule cell from all these glomeruli. You also get feed-forward inhibition onto this granule cell. And if you do the multiple glomerular stimulation, you can see this adds up very nicely. You get very strong both feed-forward excitation and inhibition. It's going to regulate how granule cells get recruited. On average, it's about a 4-fold increase going from uni to multiglomerular activation onto a granule cell, and a little bit smaller increase in excitation onto a granule cell. So this is an extreme jump into circuit stuff now from the modeling perspective. But so this is really just the larger picture of this. And so every granule cell receives feed-forward inhibition. So I know a lot of people ignore these cell types, the deep short axon cells, in their models of olfaction. I'm clearly a splitter in my models, and I've shown with this work that every granule cell in the olfactory bulb, essentially every granule cell, receives feed-forward inhibition. And interestingly, this is feed-forward inhibition that targets dendrites of granule cells, and in 10% of the cases onto the gemmules, onto the spines of granule cells. I don't know where within each granule cell dendritic arbor this inhibition is targeting. That's something that for future work, but understanding where that inhibition is targeting relative to excitation is going to have absolutely critical responses or critical impact on what granule cells are doing in the olfactory bulb. So if you imagine you activate one glomerulus with an odor, that's going to activate mitral cell A. It's going to excite this branch of the granule cell, but if it also drives feed-forward inhibition onto remote dendritic parts, remote dendritic branches of the granule cell, what you're going to get is preferential feedback, recurrent inhibition onto that mitral cell. You're going to damp down your lateral inhibition, And conversely, you can also promote preferential lateral inhibition over recurrent inhibition, and you can also generate dendritic spikes with this mechanism. And so I think I'm trying to sort of emphasize that we need to stop thinking of just granule cells getting recruited by specific mitral cells over and over the same mitral cells every time. But I think granule cell assemblies can get recruited based on the different synaptic input onto each granule cell. And so which granule cells are recruited by which mitral cells completely depends on the context, feedback projections, and also these feed-forward projections. That's why.
So I begin to learn the neuroanatomy in insects in the laboratory of Brad Smith. And so what I found that we can see all the neurons that involve into the odor transduction into the antennal lobes. So it's begin into the antennae. Antennae have a sensory receptors, which converts into the antennal lobes. And antennal lobes have two distinct glomerular neuropils. It's in insects, in honeybees, and aglomerular neuropil. Glomerular has the unit which receive olfactory receptor neurons in the certain area, and also projection neurons which connect to the higher brain order. So what I now want to learn here is about the mouse. This is my presentation of mouse. That's what I know, that they have olfactory nerve. I learned that they have mucosa. Now it's very important. And what I just also noticed from the literature is that all of these like insects, mouse and human, have the same size of glomeruli. It's ranging about from 20 micrometers to 60, 80 micrometers. And however, in insects there is one glomeruli have respond for one odor receptor. In mouse, there is about 2,000 glomeruli, and it is for 2 glomeruli for one receptor. And it's very interesting that the human has very like 5,500 glomeruli, and the ratio is 60 glomeruli for one receptor. And mitral cells, what it's estimated is in insects, it's about 5-6 projection neurons for the glomerulus. In humans, about 8 mitral cells, tuft mitral together. And however, the big difference is the chemical neuroanatomy. Olfactory receptor neurons in insects have acetylcholine, while in the mouse, it's glutamate. And what I want to know and make the mapping exactly the same way as in honeybees is all of these cells that surround in this glomerulus, what they are, and all other cells as well.
So we've been on an excursion in my lab for about 5-ish years, because a lot of the data we were collecting seemed to be quite variable depending on which student had trained the animals and which task we had chosen. So we started to get a feeling that depending on whether we did a go, no-go task or 2 alternative choice tasks, we would see different effects at the level of population activity or neural oscillations in the olfactory system. And our working hypothesis when we started was that go, no-go tasks promote beta oscillations, and go, no-go, and 2 alternative choice tasks use more gamma oscillations. So some very smart graduate students, Donald Frederick and Daniel Rojas Libano, who have now left to go on to better things, decided to take all of our training protocols and normalize them so that all of our rats in the lab are now trained in the same way for the first few steps of training until the very last step at which the 2 tasks or whatever tasks diverge. And it's very well controlled, and our animals get trained now in about a week if the students set up the olfactometer correctly. So what we found is that the two tasks produce not very different effects. They're more a matter of degree. But what we also found is that then comparing this to our previous work, it shows that there's really extreme heterogeneity in how the system is used depending on what the animal is doing. This was not news to us, but also what type of task and what cognitive factors and training factors are present in the task. So as it stands now, we now know that when we train animals to poke their nose and sniff an odor for as long as they want, they sniff longer in go-no-go tasks than they do in two alternative choice tasks, about one sniff longer, about 125 milliseconds. They perform better in the go-no-go tasks, and these differences emerge only after the point where they transfer the learned behavior to the next odor set. The first training odor set, everybody's the same. So, and what we find physiologically is that the first few sniffs, and here you see them, this black line is when the animal nose pokes. We have about a 60 millisecond delay for the odors to arrive. The first few sniffs, we get gamma oscillations, then merging into beta oscillations. These gamma oscillations, when we decompose them with wavelets, we find this typical sweep, which we've all known about for years, but which we now know is probably related to tufted cell activity followed by mitral cell activity from combination of Andreas Schaefer's lab, Fukunaga, Izumi Fukunaga did this beautiful work showing this. So what we find in our data when we do a more detailed analysis with wavelets is that we get a double-peaked gamma oscillation band. So these are collecting the frequencies of all the bursts we've detected. And we get a lower frequency, which have been tentatively assigned to mitral cells, and a higher frequency tentatively assigned to tufted cells. They occur in sequence, early to late. And then beta oscillations are a single frequency. And if we make a histogram of the of the onset of each of these types of oscillations, what we see is the onset of gamma oscillations very stereotyped. So these animals will sniff sometimes up to 15 seconds if we let them, which we do, but generally on the order of about a half a second. We see 4 very stereotyped sniffs in go-no-go tasks, the first one occurring before the nose poke. Similar in two alternative choice, only it's a little bit shorter. The beta oscillations start about 100 milliseconds earlier in the two alternative choice task. And just to leave you with one other thing is that there are we think there are multiple ways to make these different neural oscillations. And we're going to talk tomorrow about the computational work we're doing on beta oscillations. This example of a beta oscillation is not from learning, but from odor sensitization, where we give an animal a very highly volatile odor to sniff several times and can readily elicit these types of oscillations. So what we're looking at in models is multiple ways to make these oscillations.
Thank you. So we've been working on, in my laboratory, we work on the inhibitory circuits that are in the piriform cortex. And so this is just a little bit more the detail in the picture of the piriform cortex that Christiane showed this morning. And this is some of the inhibitory circuitry that exists. So here's a kind of a laminar structure of the piriform cortex. Layer 1 is where the olfactory bulb inputs come in. And there's also the source of the recurrent connections between the pyramidal cells also make a band in layer 1, so there's a lot of excitation here. And then these inputs, both the afferent input in as well as the recurrent circuitry, activates two effectively seemingly separate networks of inhibitory neurons that are responsible for things that modelers like to model, like feed-forward inhibition. That's mainly driven by the afferent drive into the network, activating horizontal neurogliaform and putatively VIP cells, although we now know that this is probably not true. It's responsible for things like sparse firing and selectivity, and this has been shown by a number of labs, but most recently in vivo by Cindy Poo and Jeff Isaacson. It also mediates dendritic inhibition, which might be important for the controlling of LTP, also shown by Stokes and Jeff Agason's lab, as well as Suzuki and Beckers, who have done actually most of the characterization of these inhibitory circuits up till now. And this is really just all the inhibition onto the pyramidal cells that people have been focusing on up to this point, as well as some early work by Haberle's lab dealing with the LTP and how dendritic inhibition might affect LTP in these circuits. And then on the other half, there's the recurrent inhibition, another thing that modelers like to model, which is really driven by intracortical drive. So it's kind of secondary to the activation of these pyramidal cells by the afferent inputs and some descending input. Deals with spike timing because a lot of this controls the somatic inhibition of pyramidal cells. And there's work done by lots of other labs on that, including Nathan Schoppe's lab. He's actually got an even more recent paper really dealing with this circuit as well. And then the overall control of network excitability also through this. This is a circuit that's apt to run if you don't keep the cortical dampening. So in our lab, what we found is we've kind of looked at these circuits as well, and what we find is with respect to these layer 2 pyramidal cells, they receive a lot of inhibition from their own local layer, and that is mediated probably by some of these feed-forward inhibitory circuits here. But what we generally find is that this inhibition is much, much weaker than the inhibition mediated by layer 3, and like 5 orders of magnitude weaker. Some of that might be because it is dendritic, and so when you're patch clamping, that's hard to see. But I actually think that this circuit is much more dominant, and it makes sense because there's way more interneurons down here than there are up here. Just straight numbers game. So we've been really interested in looking at just kind of how this inhibition onto pyramidal cells is affected through the laminar structure here. We also know that now that there's a rostral caudal gradient in this inhibition mediated by the layer 3s. So all fun things with respect to how excitation might flow through this, flow through this circuit from layer 2 to layer 3 and then along the rostral caudal axis of the piriform cortex. So we've been looking at it from this point of view, but this is really just reproducing a lot of the work that all of these other people have already done. And so where our lab is going in a really different direction is when you want to have an associative assembly form, you want to have an LTP in a circuit, you want to have information flow through a circuit, you have to tell neurons when they can fire, who can fire, and which synapses can get strengthened. And in order to do that, you need to gate some of these inhibitory pathways so that things happen at the right time and with respect to the right neurons. So we're really interested in the inhibition of inhibitory interneurons and specifically the gating of inhibition, another thing that I think modelers really like to model. And so what we're finding is that we have at least, we know of now 2 disinhibitory circuits that we've been looking at really closely. One is an interneuron targeting circuit that goes towards the somatic targeting that's in the recurrent feedback pathway. These are actually both in the recurrent pathway. And these are mediated, we're thinking, by somatostatin cells that are coming onto a generic fast spiking interneuron here. And then we also know that there's a VIP interneuron-mediated circuit that seems to gate some of this dendritic targeting circuit. We think this one is really responsible and really important in LTP, particularly because it can be activated or upregulated by cholinergic inputs to this pathway, which might be really important for learning. So I have tons of data on this, so I'm happy to get really gritty into the details in discussion sessions later. I just wanted to present this kind of generic overview of what we're doing in the lab.
So thank you. So I'm going to actually step back and talk about 2 things that I'm especially excited about, one that's been ongoing for a while now and one that we've recently got interested in. But I think I really want to start with this idea that when we talk about circuits, when we talk about networks of neurons, there's this notion about the interactions between cells, understanding the synaptic connectivity, understanding properties of the EPSPs, the IPSPs, and the computational instantiations by way of weight matrices, by way of connectivity, and so on. But the thing that I think is really interesting, and this kind of alludes to something Jim touched on, which is the diversity in potassium channels. And I think if we imagine that channels matter, then we can think of each neuron as, each neuron as an expression or an example that pulls from these different distributions of channels. And I'm just going to summarize this in the following way. This is actually some work that was done with Ken Hovis, who is a graduate student in Nathan Urban's lab. What you're looking at is an electroporation of a single glomerulus and all of the connected mitral and tufted cells. And in the green, you're actually looking at an immunohistochemical image of a single potassium channel that's differentially expressed across this population. And if one looks at these two neurons, which are these 2 points here, we see that one cell expresses the channel and the other cell does not. And it's simply that diversity that I think actually reveals a lot about the potential computational role of kind of having different combinations of channels in the brain. And a number of other groups, this is from Troy Margary's work, where they've actually recorded from different mitral cells and characterized their biophysical heterogeneity. And I think that one of the things that's really come out of a lot of this is on the left is actually some work from Leslie Osborne's lab, and what you're looking at here is actually recordings that were taken from primate, I think this is MT, and this is a visual motion area in primates, and they simply recorded from a number of neurons, and they looked at how much the diversity in the tuning properties of those neurons. These are extracellular recordings, and what they found is that actually having neurons with different tuning properties at the level of the circuit can increase the information content of the ensemble in, I think in this case, as much as twofold. And I think that's quite striking, but if one simply looks at biophysical heterogeneity, simply varying channel composition, we see that something like 20% of the information content in an ensemble of neurons can actually be explained just by channels. And the intuition there is that each channel is a different filter, and independent of the connectivity, if we have neurons that express different combinations of channels, then they're all filtering information in different ways. And I think these kinds of issues are going to become very important when one starts to think about temporal coding in the olfactory system. And the second thing, which I think is something that I've become really excited about, is actually looking at feedback to the olfactory bulb. And I think that this, for me, one of the reasons why this is perhaps really provocative and interesting is this notion that we like to make analogies between vision and olfaction, and I think that there is a tendency to find similarity, but I think if one looks through the lens of evolution and also looks through the lens of neuroanatomy, we see real distinctions, and so it may be that olfaction is in some ways very different than vision. And I think one of the reasons for me is that the anatomy of the olfactory system is actually quite different. And this is actually some methodological work that we've done using the monosynaptic rabies virus. We made injections into the granule cell layer of the olfactory bulb, and we looked at the ensemble of neurons, and we've actually done whole brain reconstruction. So we get about 1 terabyte of data out of each mouse, actually each mouse's brain. We don't care about the rest of the mouse. We can actually identify individual cells. We're working on methods to automate the classification of these neurons to identify their morphology. And one of the things we see, and I think Christiane really highlighted this, is that nearly every place in the brain that's exciting and cool projects back to the olfactory bulb. And one of the areas that we found that we're really interested in is actually monosynaptic projections from ventral CA1 hippocampus directly to the olfactory bulb. And I think that the compactness of this feedback connection, and a number of people have actually been looking at this electrophysiologically. That's some work from Venki Murthy's lab on the left and from Jeff Isaacson's lab on the right. They've really showed that this feedback can modulate the circuit in very interesting ways, and that it can have, this is from Richard Axel's lab, it can have important consequences for behavior. And I think really understanding this circuit, perhaps in the lens of feedback, may be as revealing as thinking about a system that's entirely feed forward. So, question? Do I have time? Yeah.
I kind of just brought an overview of the kind of things we have been doing and doing. So we do a lot of, I'm really interested in modulation, as most people know, expectation, modulation, learning. So do a lot of behavioral pharmacology and electrophysiology, and then pair it with large-scale simplified models of the olfactory bulb. So we've looked at acetylcholine, noradrenaline, and recently serotonin. We, postulate things like signal-to-noise ratio modulation, learning, changes in dynamics. We've also done some more abstract proof-of-concept models, mostly together with Tom, about, what's the scheme of inhibition in the olfactory bulb or antennal lobe that would be best suited to the computations that we want to perform. How does olfactory bulb modulation affect cortical readout? Questions like that. Let's see, can I do this? Yes. And so for me, some of the new things I want to get into right now, and actually whoever talked right before me said it perfectly well, is the connections between the brain structures. How are expectations conveyed to the olfactory bulb? How do expectations change what's done in the olfactory bulb? How does context affect processing, previous learning? And we already have attacked that with modeling. So this was a simple model of Natalie Mandaron's data showing that you can have purely context activation of granule cells in the olfactory bulb in absence of any stimulus. And that contextual activation can look just like the activation you get in response to odorants. And so we created a pretty abstract model of this showing that if you have LTP between piriform cortex and granule cells, that you can reproduce this data. This model had to include the very abstract notion of context cells that existed somewhere. And so I kind of want to start going after these questions a little bit more experimentally as well. And then one project that I'd really like to push a little bit is the role of adult-born neurons, their function, the rules of integration, and that kind of more long-term type of plasticity.
Thanks, Brian. I'll also keep it short because so what I thought I'd show is just a quick, first of all, a shout to modeling, and second, sort of what I think is one of the questions I'm most excited about right now. So as some may know, my take on olfactory bulb is a lot of its responsibility, its role in odor learning, which is a much larger problem than just bulb, is really about carving up an enormous high-dimensional space into something you can tractably work with computationally, which basically means categorization learning. And there's lots of details, but the approach we've taken has been to go out and do things, learn from the experimental literature, of course, do small models about particular topics, but to try and bring it into a common consensus biophysically based model where we can ask questions of the model that would really tie you into experimental data. Like are there enough dynamics, enough of the right receptors in the right places that we could say in the model, what does acetylcholine do, and have it actually predict something that means something experimentally that could be tested properly. The high points for me are whenever we can use a model like this to say something that's not intuitive and hasn't been tested experimentally and have it come out to be true with subsequent experiments, which I won't go into now, but I think I call those including NTCE and some of the feedback normalization work. But more importantly, the things in red here are things that we're interested in right now as you sort of proceed through olfactory bulb circuitry. Category learning and the effects of context, motivation, other quote-unquote top-down influences that shape what bulb does and how it contributes and interacts with subsequent processing, which I think is essential for the single major problem in olfaction as I see it, which is olfactory occlusion. I have this sort of story I end up telling undergraduate students about in Vision. You're trying to see the Mona Lisa, and you're here in the Louvre, and there's a crowd in front of you, and you can never see the whole thing because someone's always got their head in the way or waving a phone or a camera at you. What you do get in Vision is, you've got edges, right? You can say, okay, that's the Mona Lisa, and that is what's in front of it. The olfactory equivalent is if every pixel of the Mona Lisa was convolved with an unknown amount of an unknown part of this person's head or camera, giving you, under ideal circumstances, something like this, and under other circumstances, something like this. And how can the system pull out an odor, recognize an odor properly under environments like this, unknown backgrounds that occlude every single receptor, ultimately in the bulb? I think we can do this largely through categorical learning, where the problem becomes not what is this, but among what I know, what's the probability that something is here? But I'd love to talk about that in more detail, because obviously that's a long-term, a longer set of questions. The other thing I wanted to just, that is not mine.
Just had one slide.
No, well, there was a second one, but I don't really need it. In fact, I might just skip it entirely. The second one is to talk about, because we do this modeling in this biophysical modeling, and we prefer to change the model to match what we know about the system, which means a lot of analog interactions, gap junctions, graded synapses. Our models have been really constrained by sizes because they don't scale well to clusters when you have a lot of analog computations. So it's essentially a building a simulator, which we'd like to obviously make available to everybody, based on GPU computations and their shared memory architecture that will, through a sort of, we end up calling radical granularity, be able to perform and parallelize any simulation, biophysically based or otherwise, without the user needing to actually write explicitly parallel code to do this. And I have something to show you, but I don't have it, so thank you.
Okay, just moving on. I contributed 2 slides to the talk on 2 very different things, so I have one that comes at the end here. And I'm just going to try to take only about 2 minutes to describe what this says. We do a lot of bioimaging here in the antennal lobe of bees. These are glomeruli on the dorsal surface, and we can look at the spatiotemporal activity pattern that any odor gives rise to. And then because it's spatiotemporal, we can convert these using principal components and so on into these trajectories. So this is the trajectory for one odor. This is the trajectory for a different pure odor. This is a 9173553719. So as we grade one mixture into the other, you see this nice stepping across this stimulus space. There's nothing really nonlinear about it. And we're very interested in in the plasticity that you see in the antennal lobe, which you see associative and non-associative synaptic plasticity here in the antennal lobe, just like you see in the olfactory bulb piriform cortex. And we were wondering what that means for coding. So what we can do is we can compare animals that are naive to an odor, animals that are conditioned to an odor, and we can do this within animals too. This happens to be a between animal example. And if we train them that the 9-1 mixture means reinforcement and the 1-9 mixture means nothing, which the bees can solve that problem. It starts to get a little dicey for them, but they can do it, discriminate those 2 odors. And we can calculate the Euclidean distance at each step along these trajectories, each of the 100 millisecond time points as those trajectories evolve in response to odor. And what we've seen is that the distance increases in the naive animals, reaches a peak, the odor shuts off, and it comes down. However, in the animals where they've been conditioned, the distance between those trajectories increases much more significantly so. So what we're arguing is one of the things that plasticity is doing is it's reshaping temporarily, at least, the coding space to stretch a perceptual dimension that represents these 2 odors and make them, at least while the problem is current, more detectable and discriminable. We've done some modeling starting with Christian many years ago and also with Ramon and Maxime now. And this is something Christian and I published some time ago. And nope, it goes away. And what I want to emphasize is that in all the modeling, it points to the importance of modifying the inhibitory circuitry in the antennal lobe or the olfactory bulb. So it's the inhibition where a lot of this plasticity that drives this kind of recoding takes place. Anyway, let me end it there. I promise to be short, and we're late. And we'll move on to our last talk, and if I can, by way of just a brief 10-second or 15-second introduction, is Gordon Burkhart, who's a native Tennessean, or at least from here.
No, I'm from Wisconsin. Nobody has ever pitted me as a
Okay.
Tennessean when they hear me.
And one of the things that has not come up yet today experimentally, because I think that virtually everybody, aside from a few mentions of honeybee, we're talking about rats and mice. And something that might warrant further discussion is what about other animals and their olfactory systems and what they can possibly teach us. And I think that's what Gordon's going to address.
Okay, I'll be fast, and I'll read this sort of. But snakes are really perceptually unusual. They are the most chemically dominated vertebrates. They use their tongue flicking system. So when the snake flicking its tongue, it is actually transferring primarily, but not exclusively, non-volatile chemicals through the vomeronasal organ, which I'll show you a picture of in a second. It's in the roof of the mouth. Rats have these. Many mammals have it, but snakes have perfected the use of this system. And it's used in recognizing and trailing, avoiding prey, anti-predator behavior, finding mates, recognizing siblings, habitats, and so on. Many snakes are also extreme dietary specialists, and this is where the issue of evolution and comparative studies, I think, is something that snakes really can provide, because they have very precise and specific dietary preferences of foods, even within the same genus of animals. And let's see, how do I transfer?
Just on the keypad, on the computer.
Oh, okay. So garter snakes also are very unusual, are very useful because they have large litters. You can have 30, 40 animals from one mom. So that means you can control for genetics, and we've also shown they have multiple paternities, so we can identify who are full sibs and half sibs and so on. It's the largest genus in the Natricinae, which is a really useful study species. They're the most widespread snake. They live in very much in diverse habitats. Even between Wisconsin and Michigan, for instance, there are the baby snakes respond differently to different prey odors. They respond differently to predator behavior. So there's microevolution going on that you can study. You can easily study these in captivity. I can screen animals, dozens of animals in a day on a variety of different odors really efficiently, and use the tongue flick rate, and actually also response. Okay. This particular species, as I said, is widespread, and just like naive neonatal garter snakes without any experience will respond with increased tongue flicking and an actual open mouth attack behavior to prey odors or chemicals, I should say, from respective prey. So if they may be worms, it may be fish, maybe amphibians, and so on. However, they may be very specific at birth, but they can be modified with one trial under some circumstances. Okay, and so you can enhance it. This is also true of anti-predator behavior. There are these very stable species, population, individual differences. Even within the same litter, I can have what I call chemical preference polymorphism. Some animals prefer fish odors, some earthworm chemicals, and they're very stable within the same litter. And I interpret that in terms of the mom bet hedging, in terms of environments that can change. If they get drier, they may be more prone to have earthworm or fish or amphibian or whatever prey available. These animals also differ a lot morphologically. And just to show you, this is the vomeronasal system. It has tongues flicking of the snakes. It's in the roof of the mouth. We did some first brain imaging, as I believe, of a snake. And vomeronasal organ is mushroom sort of body right here. And this is a snake attacking a cotton swab that just has the odor of earthworm in it. I've never seen it. I've never eaten anything in its life. And this is these are water snakes. They're closely related to the garter snakes. But I just want to show you this. This is an old data set showing that the common water snake that we have around here responds a lot to frog and fish odors, not so much to crayfish odors. But there are two species of crayfish snakes, they're called, that specialize on crayfish, and in fact, they can distinguish at birth odors from hard-shelled crayfish from soft-shelled crayfish. Soft-shelled crayfish being a lot easier for them to eat, right, than hard-shelled ones. However, there are related animals that we've studied more recently that prefer the hard shell over the soft shell. And there's also maturational changes. So in some species, you actually have a shift of prey preferences as they grow older, which can be interact with experience or be independent of experience. So I think snakes really offer a lot of really neat opportunities. We've also done physiological recording and so on, so lots of things about snakes. I hope you may think that snakes may be useful for some kinds of modeling and also experimental work.